everyone and welcome to this class. I welcome you all to Science 101 PG channel. So, in today's topic, we are going to learn about the pathogenicity islands. Now, it can be a new topic to someone and it can be old topic, but we are just introducing in 101, 101, whatever you want to say, or in quick fashion. Okay, so let us start with what are these pathogenicity islands. Okay. So pathogenicity islands, or in short, I am going to call it as PAIs from now. So basically, these are genetic elements. Genetic element basically means DNA. Okay, because we are going to talk in case of bacteria most, we are going to use the term DNA, not other things. Okay, and basically, if you talk about RNA, then RNA are only found in viruses or lower categories of subcategories. Fine. So as I was saying, PAIs or pathogenicity islands are the genetic elements that are found in chromosomes of many bacterial pathogens and are associated with virulence. Okay, don't forget this term virulence because just keep using it in PAIs. It will help you. Okay, so what are these PAIs actually? So if you talk about PAIs, then these are the segment of DNA that are typically acquired through horizontal gene transfer. Okay, now this is the first bacterium, or you can say some pathogenic bacterium. So what will happen is there is horizontal gene transfer. This is horizontal. Okay, so horizontal can be transformation, transduction, or can be conjugation. Okay, so these three are basically horizontal gene transfer method. So this gene which came from the pathogenic bacteria will go and enter into non-pathogenic bacteria or simply some other bacteria. Okay, and this will convert this non-pathogenic bacteria into pathogenic bacteria by increasing its virulence. So that is the basic mechanism behind the pathogenicity island, okay, or PAIs. So what are the benefits of PAIs to bacterium? So if we talk about bacteria, then if PAIs are being introduced into one bacterium, then it is providing tools necessary for the colonization and invasion and evasion to the host immune system. Okay, so if PAIs enter into new bacterium, its colonization will increase, its invasion to new host or same host will increase, and its evasion to the host's immune system will also increase. Like if you take the example of macrophages, okay, so macrophages basically will go and eat up this bacterium, but if it has PAIs, pathogenicity islands, then there will be some release of proteins, toxins, some chemical mediators which help evade that bacterium from this microorganism. So the bacterium will ultimately save itself and move away from the bacterium. Okay. So this is the basic idea if you talk about PAIs. Okay. Okay. Let us move to the next topic, which are what are the characteristics of PAIs, pathogenicity islands. If you talk about the characteristics of PAIs, then first characteristic is going to be the point regarding the genomic islands. Okay. So PAIs are basically subset of genomic islands. Now, if you don't want to say subset, you can always say a part. Okay. So what are genomic islands? You might be curious. So here is your answer to that. That genomic islands are large segment of DNA that are mobile. Basically, if you talk about genomic island, then as I am saying that PAIs are horizontally transferable, so are your genomic islands. Okay. And if you talk about the size of PAIs, then their size varies actually, and it typically ranges from 10 kilo base pairs to 200 kilo base pairs. Okay. Then the third point will be your GC content, point of GC content. Okay. So first of all, if I talk about any genetic material, if this is the genetic material of your bacterium, then the GC content is kind of fixed if you talk about same species. But when the GC content of PAIs or basically PAIs will enter into PAIs. When the PAIs will enter into the and mix up with the genetic material of bacterium, then the GC content will be different, right? So here is your third point regarding that usually their GC content is different compared to the original genome of bacterium. Thus, thus this shows foreignness. Okay, basically this means that the PAIs are not actually the part of that bacterium or microorganism. You can simply say. Okay, and the fourth point is regarding the mobility. Okay, so if you talk about mobility, then PAIs contain mobility genes, just like genomic islands, because genomic islands are mobile. So are PAIs because they are again subset of the genomic island. So basically, PAIs can contain integrase, transposases, then also contain some insertion sequence that facilitate the integration and excision from the genome. That is your fourth point. And if you talk about fifth point, then in fifth point, we are going to talk about virulence of the gene. So PAIs carry multiple virulence genes that can encode toxins, adhesion genes, which can help in adhesion of the bacterium to the host cell. Okay, then it can contain many secretion genes, which can help in secretion system genes. Then immune evasion proteins, as I have given you the example of macrophages. Okay, it is also there. Also, apart from this fifth point, you should also remember that PAIs, I'm going to write it as sixth point. So, PAIs have repeated nucleotides. NTD, it means nucleotides. Okay, nucleotides on both hands. On both hands. Like if it is the gene of, or you can say if this is the PAI, then it will end like A, T, 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 or instead U, whatever. Okay. So these repeated sequences are also found at the ends of the PAIs, Pathogenicity Islands. Now here is the next point, which is origin and adaptation. Okay, these two are common. Okay, so if we talk about origin, then horizontal gene transfer uh, can be can be said as the origin for the PAIs in bacterium. Okay, if you talk about horizontal gene transfer, as I've told you earlier, transformation, transduction, and conjugation are the three modes by which they can be transferred from one microorganism or one bacterium to second, and so on. Okay, then we have the point of plasmid phages. Okay, so basically plasmids and phages are mobile genetic elements. Okay, and they often carry PAIs. So that is also a method. Like if you talk about plasmid, then you have sigma replication. Okay, if you talk, and sigma replication can transfer the plasmid from one bacterium to another, and similarly PAIs, if it contains PAIs, then this can also be transferred. Then your adaptation feature will be the evolutionary adaptation. Okay, now this point is very important because as you can see that adaptation to PAIs lead to adaptation to new host. This is not the case for all the time, but sometimes it happens that. Okay, so why I have written adaptation to PAIs. Okay, so let me say that this part of segment is a PAI. If it enters into the bacterium, it has two fates. The first fate will be that it will survive. Okay, and the second fate will be it dies because of like nuclease activity or some other activities, right? So if it survives, then only you can say that the bacterium adapts to the PAIs. And if that PAI gets integrated into the genome of bacterium, then it can lead to the adaptation of new host. Okay, let us say the PI encodes for some protein that have affinity with some receptor on monkey plus human. Okay, so earlier if that that norm I mean that bacterium is affecting a mammal, then now after the introduction of PAI, now this bacterium can also infect monkeys and humans also. So that is how the PAI, you know, show the evolutionary adaptation feature. Now, if you talk about the examples of PAIs, then first example will be the example of your salmonella. Now, in salmonella, we call them salmonella pathogenicity islands, or in short, SPIs. So, you will get to see two SPIs. First will be SPI1 and second will be SPI2. In SPI1, SPI1 contains the gene for type 3 secretion system, which we call as T3SS, type 3 secretion system. Okay, and this TS, T3SS will be responsible for invasion of bacterium into host, and that to facilitate the process of invasion. Okay, so if you talk about SPI2, then this SPI2 will encode for another type of T3SS or type 3 secretion system, which is involved in survival and replication within the host macrophage. Okay, so basically this make it as evasion type. Okay, now in second case we are going to study about the case of E. coli, in which we call them uropathogenic E. coli or UPEC in short. So, okay, so pathogenicity islands. So in E. coli, you will get to see PAI1 and you can also see PAI2. Okay, so PAI1 contains the gene for hemolysin production and adhesion. Okay, okay, hemolysin production and adhesion. Okay, so these two are very important for E. coli if E. coli is going to infect the urinary tract of humans. And if you talk about PAI2, and this encodes for fimbriae on the surface of E. coli. So basically it will mediate in attachment or help in attachment. Now the third and the last example of this video will be your Vibrio cholerae. In this you will find these two kinds of PAI, which is VPI1 and CTX-PHI. Okay. So in VPI1, which is Vibrio pathogenicity island, then this please remember this, then that this contains TCP, which is toxin coagulated pilus. Okay, so this is a kind of operon which is essential for intestinal colonization. And similarly, you will have CTX-PHI, which contain cholera toxin genes. Yes, you are right, folks. Cholera toxin are not found in each and every Vibrio cholerae, but in some pathogenic strains of that. But if CTX-PHI pathogenicity island are transferred from pathogenic bacterium to non-pathogenic bacterium, then the non-pathogenic bacterium can again become pathogenic. Okay. So now we will talk about the functions of pathogenicity island or PAIs. Now I am going to use these three examples to demonstrate the pathogenicity island. So the first will be your colonization. Okay, here VPI1 contains TCP operon essential for intestinal colonization. This is your first function of pathogenicity island. Okay, then you will get about then you will get the information regarding the invasion. Here invasion SPI1. SPI1 contains gene for type 3 secretion system for invasion. This is your example for invasion. Then you have evasions, as I have told you right now that evasion from macrophages. Okay, so here it is the evasion SPI2 encodes the another type of T3SS type 3 secretion system which involve in survivor application within host and evasion from macrophages. Okay, then you will get to see about toxin formation also. It was of Vibrio cholerae CTX-PHI, CTX5, which contains cholera toxin gene CTX-AB. Okay, so these were your functions of pathogenicity island PAIs. Now we will move to the is it last? Yes, this is last. Okay, so the last topic of the day will be clinical and epidemiological significance of PAIs. Okay, so of course we have read about how dangerous these PAIs can be. because it can convert a non-pathogenic microorganism into pathogenic, okay, by introducing some foreign genes into the non-pathogenic bacterium. Okay, so first will be the case of disease surveillance. Okay, so if one person can monitor the PAIs, then this can help and track the virulent strain in population. Okay, so PAIs being special gene, if one is able to track that gene or PAIs, then of course it will help track the virulent strain in population. Our second point regarding this, wait, second point regarding this will be vaccine development. Okay, now as you know, after the COVID, the importance of vaccine is just too much. Okay, so now regarding the PAIs, what happens is if we target a certain PAI encoded gene, targeting PAI encoded gene. Okay, I'm sorry. I must have written in it in some bad way. Okay, let us say this is my PAI gene and it encodes some protein, right? So this can lead to the development of new vaccines against that protein. Okay, and this can help develop new kinds of vaccines so that in case of emergency we must have some stock with us, right? That is how vaccines are developed by using PAIs. Then our third point will be antimicrobial strategies. Okay. Now understanding of PAIs, pathogenicity islands, can lead to the novel antimicrobial therapies which can disrupt the virulence of the microorganism. Okay. So this is also a very important point if you consider about uh, clinical and epidemiological significance of PAIs. So that's it for this video. Thank you very much and see you soon. Of course, before leaving, please don't forget to like, share and subscribe to my channel. Science 101 PG, don't forget. Thank you very much and see you soon.